Genes predictions in eukaryotes. There are different tools which are used for gene prediction in eukaryotes. Most common tools they are Genie, Fgenes, and Genscan. So these are three most important tools which are used for gene predictions in eukaryotes. Gene prediction tool, that is Genie. The model includes the information that average length of exon and intron and compositional information about exon and intron. In addition to these two, that the length of exon and intron and the composition of exon and intron, a neural net derived model of splice junction and consensus sequence around splice junctions are used by this tool. Genie is approximately 60 to 75% accurate on eukaryotic genomes for identification or for the prediction of genes. Here is the example of gene prediction in eukaryotes, that this is the actual gene structure where we can see that this is exon, and next exon. Here is the start codon and here is the stop codon. With the help of Genie, initial prediction can be in the form of that exon 1 and some other exons can be identified by the Genie. So here we can see that initially it has identified 5 exons. But by performing the sequence homology alignments, the irrelevant or the wrong identified exons can be eliminated. So here we can see that by sequence homology alignments, the exact exons have been identified. So this is the tool which is called as Genie, is used for genes predictions in eukaryotes. There are some advantages and disadvantages that extra predicted exons can be eliminated based on the evidence from homology searches. And on the other hand, it works best only for the mammalian genomes, not for other eukaryotes. So these are some of the advantages and disadvantages of this tool. The second tool, which is called as Fgenes, is used for gene predictions in eukaryotes. This software identifies the exons and introns and gives the score to each exon and intron based on the composition, and uses the dynamic programming to find the highest scoring path through exon and intron, and ultimately the best scoring path is constrained by several factors, including that exons must be in frame with each other and ordered sequentially. So this is the methodology that how Fgene works for the gene predictions in eukaryotes. Genes predictions by Fgenes. Fgene is approximately 70% accurate in case of mammalian genomes. This is the diagram that how Fgene identify the genes. That this is the actual gene structure. This is exon 1, exon 2, and exon 3. Initial prediction with the help of Fgenes can be that it gives the score, that here it have given 0.8 score to exon 1, 0.3 to another exon that have been identified by Fgene, then here another exon 0.94 and 0.92, then 0.5. So based on the scoring system, the actual exons can be identified. Here we have seen that here it has given the score 0.3 and here 0.5. So it may be the false exons. So as an initial prediction, it was actually this was exon 1 and exon 2 and exon 3 of the gene. So final gene structure prediction, that this is exon 1. So these are the wrong identified exons because the score was 0.3 and 0.5. So Fgene is another tool which is used for the gene predictions in the eukaryotes, which is based on the scoring system. There are some advantages and disadvantages that alternative gene structures are reported, and only human model based version is available for unrestricted public use. So these are some of the advantages and disadvantages of tool which is called as Fgene for the prediction of genes from the eukaryotic genomes. In addition to these two, there is another gene tool which is called as Genscan. Genscan models for different states. There are 3 different states, state 1 and state 2, that is for the exons and introns, to identify their length and the composition of the nucleotides within the exons and introns. And in case of state 3, it also checks the splice junctions of exon and introns, which are also called as boundaries of exon and introns. So in case of state 1 and 2, it identify the length of the exons and introns and the base composition of the exon introns. And in the third state, it also identify the splice junctions. Weight matrix based array to identify consensus sequence, weight matrix to identify promoter poly A signals and other features, means with the help of Genscan, it also scans about the promoter sequences as well as poly A tail sequences. What are the outputs of Genscan tool? It identifies the gene structure. It identifies the promoter site, translation initiation exon, internal exons, and terminal exon mean translation termination exons. Genscan is approximately 80% accurate on human sequences. There are some advantages and disadvantages. Most accurate of available tools, but user cannot train model nor weak parameters. So this is the comparison between the advantages and disadvantages of the tool which is called as Genscan, which is used for gene predictions in case of eukaryotic genomes. So gene prediction eukaryotic Genscan, eukaryotic gene structure can be quite complex, as we know that there are exons and introns. In addition to, there are other sequences which are present along with the genomes that can be promoter sequences, that can be enhancers, silencers. The best approach to gene finding in eukaryotes is the combination of different methods. So for identification of genes or for gene predictions in case of eukaryotes, that more than one tools can be used for the prediction of the genes in case of eukaryotes.